been working on. And a couple of years ago when I did my first faculty seminar, I dressed up like Clint Eastwood, and I did the good, the bad, and the ugly of marine viruses. And since then, times have changed. I'm not sure how I feel about bringing a shotgun into like a Florida classroom. So I decided I'd be a little mellower today. And I'm going to tell you about basically our mystery cases. And I did bring a costume because I didn't want to disappoint. So I have this like awesome Sherlock, which is maybe backwards, I don't know, and my pipe. But I didn't think through how I was going to have a seminar with this in my mouth. So you'll just have to go with it. Okay, so
you can use it to point.
I can point with my pipe. There we go. Don asked if it blew bubbles, which I thought would have been an even better way of doing this. So my lab's a bit of an outlier here on some levels because obviously we pretend to be marine scientists, but overall we're really microbiologists and virologists. And what a lot of us are working on is really just trying to describe the diversity of viruses present in a lot of different environments, whether we're thinking about pathogens or normal healthy things that are out there. And this has kind of earned us this reputation of being virus hunters, which is the media's like favorite thing to call us. And kind of why do we care about viruses? So I have this model of a virus which I brought last time, but you can see it here, and it's something that they sell to kids as toys. And this model, I think, is this great description of viruses because it calls them tiny bundles of evil. And then in the instructions, as you read to assemble them, because if you have a PhD, assembly of these things, like if you're 7, it's really easy, apparently. If you've had 7 years of school under your belt, not much. So they call viruses mobile modular munitions plants for producing weapons of micro-destruction. They call them biocrystals. So they're really small, but they are biological. There's a lot of debate and controversy over whether viruses are alive, but regardless of whether you believe they're alive or not, they can really wreak havoc, right? You know, if you've ever had a flu, for example. So these biocrystals are smaller than a speck of dust and inert as a rock until it drains your life force to clone itself with robotic precision. And really these viruses, despite their small size, so the model that's about this big is, you know, a 10 million times magnified, but these really small things can really wreak havoc on everything from our bodies to whole ecosystems. And viruses are really tricky to identify, which is where we kind of get into the detective part of this story. So a lot of organisms have conserved genes that are found in all of them. And so it's pretty easy if you want to be able to identify like a bacterial infection. But with viruses, they don't have anything that's conserved in all of the genomes. And they can have really different types of genomes. So they can have DNA genomes, they can have RNA genomes, they can live in lots of different places and really be specialized. And so we don't have one overall test that tests for a viral infection. And you know if you go to your doctors, right, you've got a cold, maybe, or maybe it's a virus, maybe it's a sinus infection, right? Usually when you go to your doctor, you're like asking for antibiotics. That's what most people want. And if you have a virus, antibiotics aren't going to work for you. There isn't like a quick, easy way that the doctors can just say, this is a virus. They have to actually, if they wanted to, which they're not going to do in a normal situation, test for each individual virus that they think might be involved. And so we have different tests, PCR, which uses a specific gene sequence that is unique to a virus, or immunological assays that look for antibodies to test exposure to different viruses. We can also see them under microscopy, but they're very small. We can grow some of them in the lab on cell lines, but it means that we have to have all the right cell lines, and even with that, we can't culture a lot of the viruses. And when we're talking about marine organisms, which is what I'm going to focus on this morning, you know, we just don't have good cell lines and immune assays for things like sea lions or sea turtles, and it gets even worse when you're thinking about something obscure like a viral infection in a copepod. And so if you watch a lot of TV like me, it's not just Sherlock I'm obsessed with, it's also House, because this is, you know, a show where people come in with really weird diseases and he like does all this stuff to figure out what it might be. And the real problem with what we do is how do you find something if you don't know what you're looking for, right? So if you come to me and I suspect you have a rhinovirus, I have a myriad of tests that I can do for that. And I can either say yes or no. But if it's a no for that, then I want to move on to the next most likely virus, right? And then the next most likely, next most likely. And actually you have to test for each of these things essentially individually. And so what my lab is working on are techniques that let us sort of see what's there as opposed to running through all these individual tests. And just like Sherlock has his Dr. Watson that helps him through with everything, we also have a very helpful Dr. Watson, but it's a different one. And this is Watson and Crick, as well as Rosalind Franklin, whose birthday was just like 2 days ago, who really figured out the structure of DNA, right? And so we've learned a ton about nucleic acids, and we use all of this information that we know about genomics to find viruses. And the methods are pretty simple and basic, at least on the original side. So mostly what we do is grind stuff up and sequence its DNA. And it's a little bit more complicated than that, but I know this is what Gary thinks I do all day anyway, so we're good. And when I say stuff, that's the camera, exactly. So yeah, don't show this one to the Ayacook people. Invertebrates mostly. But we really can start with almost any starting material. And so we work with all sorts of stuff in my lab, anything from animal tissues to whole invertebrates, insects. We've worked with everything that you see here. I'm going to tell you a bunch of the different stories today. Overall, I have a lot of people in my group that are working with various things that I think can be encompassed with this. So today I'm just going to talk to you about active infections that we've looked at in marine organisms. But we also have a lot of work looking at sewage and how to detect fecal pollution, environmental reservoirs of different viruses that can infect humans and other hosts. And then we've also applied the same techniques I'm going to talk about today to look at insect vectors like mosquitoes and whiteflies that transmit a lot of viral pathogens as kind of a way of getting a survey for what's circulating in a given region. So this is really how the method works. It's our 4-step process. So we start with a sample, and again, this can be anything. So if it's seawater, you can just start with a liquid. If we have something like an animal tissue that we're working with, we first have to like grind it up and get it in kind of a liquefied form. Then we purify the viruses, and this step's really important. So let's say you take a tumor from a sea turtle. If you were to just sequence all the DNA from that, 99.99999% of what you would get would be turtle DNA, right? So we want to see the viruses that are there. So what we do first is purify all the viral particles out. So we're looking for those virus particles. We can purify them based on their size, based on their density. If we treat the sample with nucleases that chop up DNA and RNA, it gets rid of everything free, but the virus particle is protected from that. So we purify the viruses from whatever sample. We then extract out their genomic material, DNA and RNA, and then we amplify that fragment, sequence it, and compare it against databases, try to figure out what we've got. And this is really different than that one-at-a-time search for different viruses that I was talking about before, because it's not just giving you a yes or no answer for a different thing, but it's also can show you things that you might not have thought to go looking for, right? Including new things or things that are variable enough from the known versions that we might not be able to pick it up with an assay based on the known sequences. So just a couple of the things I'm going to tell you about this morning are some of the work that we've been doing in sea lions, as well as looking for a cause in a sea star mortality event that's been going on the past few years. And then I also want to talk a little bit about what is healthy. And Ernst used clean, I think, in your samples, sort of describing these clean samples, but it's the same concept that I'm talking about today. You know, how do you know if something is truly healthy or unaffected, and what's that baseline out there that you can compare disease to? So a lot of these things happened really serendipitously, right? So I think as a researcher, it's often hard to predict which direction you're going to go with things. And we knew that we had this method for identifying viruses, and we sort of put a very broad call out to vets, and we said, if you have a disease and you don't know what causes it, we'd like to look for viruses. And I didn't know if it was ever going to work. And with this sample in particular, I didn't think it was going to work. I was really pessimistic about it, and it worked perfectly. So it's a wonderful story to tell you. It's not all failures. So we got contacted by the lead vet at the Kansas City Zoo, and he was going through a big problem with his California sea lions. So within a relatively short period, they had 3 of their California sea lions that died. I have all kind of the medical terms up there, but it basically had lung diseases, and they couldn't figure out what it was. They ran through kind of all of the standard tests that they had available. So they checked for, chemical toxicants in the environment and everything like that. They tried to culture bacteria. They tried to culture viruses, fungi. They looked for West Nile virus and other things that they knew were in the area, and they just kept coming up blank. And so what they sent us were lung tissues that were dissected out of these sea lions after they died, and we put them through our method. And what we ended up finding was a new viral genome. That's just a schematic of the genome. It doesn't really matter that much what the genome looks like, other than that this was basically really different from anything we'd ever seen before. So these are really tiny viral genomes. They're single-stranded DNA. They're only about 2,000 bases, so they have like a handful of genes in them. It's kind of crazy to think that something that small and simple could really take down an animal, but it absolutely can. And we were really interested in these. This is a group of viruses called the Anelloviruses, and Anelloviruses are really widespread in human populations. So in some countries, up to 90% of the people can be infected by Anelloviruses. Probably half of the people in this room, your blood samples would be positive for Anelloviruses, and they've never actually been linked to any sort of pathology. So they're kind of the closest thing we know to a commensal virus in humans. And there's a lot that's not understood about how that immune interaction goes on. But these had never before been seen in marine organisms. And so the one that we found, within one of its genes, so not even with the full genome, but in one of its genes, it had about 30% amino acid identity, not even nucleotide identity, to a cat and a human Anellovirus. So this is like a very, very weak similarity, but it's enough to suggest that it's in that same family of viruses. It has a lot of conserved features. We then did a lot of follow-up work, because just because you find a virus in a given sample, right? This sample was a dead sea lion. We found a virus. It's really important to never jump to that conclusion that, therefore, that virus killed that sea lion, right? Like, what if it's something that's in all sea lions? So we did as much as we could to see if we could associate it with the mortality event. We tested all of those sea lions that died of the mortality event. There have now been sea lions from the same zoo, and they've all been positive for it. We also got samples from stranded live sea lions, or sorry, stranded dead sea lions, but wild sea lions that had died, and we found it in about 11% of those samples. We hadn't been able at this point to test any live sea lions because we could only detect the virus in the lung tissue, so we couldn't detect it in the blood at all by PCR, and no one's letting you lung biopsy wild protected marine mammals, trust me. We also were able to find the replicating form of this virus. So I told you it has a single-stranded DNA genome. When it replicates itself, it makes a double-stranded DNA form. And so we were able to detect that as well, which suggested it was actively replicating. We've also now found a related virus in harbor seals where it doesn't seem to be associated with disease. But there's still a lot that we need to do to better understand this virus. And the fact that we couldn't find it in the blood really limited what we were able to do. And so in comes one of my current evil scientists, Liz Fossbender, and she kind of picked up on this genome discovery, and the idea was, well, now can we detect antibodies to it? Because in the blood, you should be able to detect antibodies, which would tell you whether the animal currently is affected with the virus or also whether it's seen it in the past, right? So whether it's had past exposure. And we've done this working with John Cannon and his group over at All Children's Hospital and USF Health. And the idea is that we're looking for antibodies within the sea lions that are going to show us whether they've been exposed to this virus in the past. And the results are really complicated and not at all straightforward, unfortunately. But what we find is that we see some animals that are PCR positive in the lungs, which suggests an active infection right now. We see some that are PCR negative in the lungs. This was in 100 paired lung blood samples from dead animals. And those ones maybe were exposed in the past, but don't have a current infection. We see some population that's overlapped between these two assays. And then we see that about half of the sea lions are completely negative for either exposure or for current infection. And we're really excited about this assay right now, even though the results are not completely straightforward. It's hard to set this up it's actually hard to do any of this, I'll be perfectly honest with you, without like proper controls and things, right? So we don't have this virus in culture. We don't have good cell lines for sea turtles. We don't even have good immunoglobulin for sea lions. So it's pretty tricky. But one of the things that we can do now that we have an immunoassay for this is we can actually take blood samples from healthy sea lions, and we can do this with the ones in the zoos, and then follow them over time. And when they begin to show symptoms of disease, we can look to see if ones that were negative for exposure to this virus have now seroconverted to become positive for this virus, which would help us implicate the virus in the disease. One of the things that the zoos also really want to be able to do, they get contacted to take in rehab animals. So for example, animals that have stranded, so they want, they're always asked to take new animals into their facility, and the vets would really like a good way of screening those animals to know whether they might have this virus before they can let them in. So this is kind of a work in progress, but I think it's a good story because it shows you all the steps kind of all the way through, even though we're not done yet, from initial sequence detection to understanding what it's doing in the animals. The other project that Liz primarily working on is looking at sea turtle fibropapillomatosis. So I know there's a lot of new students in here, or maybe new to Florida. This is a disease that's plaguing sea turtles all over the world, but we actually have one of the most affected populations here in Florida in the Indian River Lagoon. So sea turtles get these tumors. I don't know how well you can see it, but this one has tumors on its face, also on its flippers. These tumors can grow to be really massive and to interfere with their vision, with their feeding, with their locomotion. They can also get the tumors internally, and when they get them internally, there's usually kind of a threshold once the tumors get so bad that the animals actually die from it. There's a lot of evidence that shows that viruses are the causative agent of FP, which is fibropapillomatosis, so I don't have to say it 10 times fast. So kind of back in the days when you could do these experiments, people had purified the viral fraction just by size and density, all the things that we do, and then added that back to turtles and shown that that fraction could actually confer the disease. And then in particular, there's a herpes virus where we consistently find that herpes virus associated with the disease, and it's not found in turtles without the disease. So even though we can't sort of fulfill Koch's postulates and do all of the traditional things to link a virus with the disease, because no one has been able to grow this in cell culture despite several PhD dissertations dedicated to this topic in other labs, we're pretty confident that this virus is associated with the disease. So our lab is interested in sequencing the herpes virus, but also in seeing what other viruses could be there. And so when we originally started, we did our usual method that we do. This is just to kind of show you that purification in pictures. So this is a turtle tumor all ground up and everything, and then we add a dye that binds to DNA and RNA. This lets us see DNA under the microscope, basically. And you can see there's kind of a complicated sample here. And then once we purify the viruses, we're left with what this actually looks really nice to me, but a really kind of nice sample where these little pinpricks of light are virus particles. And then we can verify based on DNA what we've done. And so in this sample, when we do a PCR to detect turtle DNA, we get a really bright band because there's tons of turtle DNA there. Once we've purified our viruses, that band goes away, or it essentially goes away. So we've done a good job of purifying the viruses. When we sequence viruses from this sample, we also found a new genome. This is actually not totally unrelated. It's kind of similar in structure to the one we found in the sea lions. But this one has a lot of different characteristics. We were actually able to name this virus. We called it a Tornovirus. And this is probably the first representative of a new viral family. I'm going to come back to the herpes virus. This one is not related at all to the herpes virus. One of the coolest things with this genome is that we didn't just find one virus genome. We actually found a whole family of related ones. And this is something that people in that field called quasi-species. And what we have is that most of this genome is completely conserved. But these are all different variants that we were able to isolate and sequence. And they have this one region that's really hypervariable. And actually every distinct genome that we sequence had a different variable region. And this is something that is thought in persistent viruses to actually, it's kind of a reflection of this arms race that's constantly going on between the immune system of the organism and the viruses that are infecting it. One of the disappointing things about this project, we were really excited that we found this virus. We went back and tested hundreds of sea turtle samples. It didn't actually seem to be associated with the disease at all. So we found it in very few turtles. So it's still important knowledge. Sea turtles are endangered or threatened, but it really didn't give us very much insight into this disease. we were pretty disappointed that we did all the sequencing and we never came up with this herpes virus, right, that we know is present in these samples. And if you remember, our first step is purifying those viral particles, right? So that requires that the virus is present in particles. And if you know anything about herpes viruses from your lives, and I'm not implying that you have herpes, but many people in the human population do. So herpes viruses are really tricky because they can undergo these different lifestyles where they go through a latent infection where you're maybe not showing any symptoms, and then they can switch into an active infection. This is actually known to be correlated with things like stress. And so herpes viruses, when they're in this latent infection, they won't actually be present in virus particles. So you'll just have the DNA either integrated within the host genome or maintained just as kind of an extra circle within the genome. You can think about it that way. And so we weren't recovering them because we were actually losing them by purifying the viral particles, which is a very important sort of management aspect in this disease. So Liz is actually working on this for her PhD right now, and I don't have much data to show you yet, but we've now got almost complete genomes of the herpes virus from 3 different sea turtle species, and we're learning a lot about this latency and how that possibly controls development of the disease. They also see regression sometimes, and we're hoping that this will help inform vets who are caring for the sea turtles who can treat with antivirals sort of because empirically it works, but we don't know a lot about, when we should be treating and how the different stages of the tumors are developing. Just a really quick story, because this is one that we got pulled into, but it wasn't our primary thing. There was a giant mass mortality of sea stars happening. Did you guys hear about this in the news? Anybody? Yeah. It was pretty widespread, so it affected tens of thousands of individuals. It was in the Atlantic, Pacific, Mediterranean, lots and lots of different species of starfish. And this basically, the stars started off looking healthy a week. They went from healthy looking stars to getting white lesions to completely dissolving. And this was really widespread mortality for a while. We didn't know anything about the cause of this disease. Working with Ian Hewson at Cornell, Liz worked on this disease, and we were able to identify another new virus. This one's a different family, but basically we did the same thing with this system. So we were able to show that the virus fraction, purified viruses, could recreate the symptoms of this. As we got higher viral loads of this virus, the disease progression worsened. And we were actually we could go back to museum specimens that had been preserved from a long time ago. We were actually able to detect this virus as far back as 1942. And this is one of those interesting things to think about when you think about health versus disease. Do you have introduction of a new pathogen, or do you have a pathogen that's been around for a while, but now it's either expanded its range or the animal's immune system has changed in a way, maybe due to extra stressors that make it more susceptible. And one of the things that we saw was that even though we saw more viruses in the disease species, which are just the red bars here, we also saw quite a lot of these viruses in healthy specimens. And when we got to the end of this project, basically our